Hello, this is Dr. B, and we are looking at the activity 4, lesson 2.1. You can find it on the menu here for deer mouse fur color from the field to the beach. And of course, we're working on iPads, so we're working on the touch device version. In Unit 1, you figured out part of the reason for why mice have different fur colors in different environments. In this unit, you'll look at two subspecies of deer mice, the field deer mouse and the beach deer mouse. The field deer mouse tends to live inland on dark soils and has dark brown fur, while the beach mouse lives on light sand beaches and has light brown fur. This unit will investigate why and how field mice have dark brown fur and beach deer mice have light brown fur. Imagine you could zoom in and look at the cells that make up a mouse's body. What might you find in the cells that could explain why the beach mouse and the field mouse have different fur colors? So think about what kinds of things in the field mice and the beach mice cells could be making their fur different colors. Take a guess. It doesn't matter. This is just a hypothesis, a first pass, a first draft, if you want to think about it that way. And so pause the video and write your answer to question 1. So now that you finished question 1, we're going to kind of zoom in here. Below is a series of images that will start with the deer mouse and gradually zoom in. Here's the skin, here's a single hair shaft, and here's one of the cells in that hair shaft. You can see those little brown dots right there. The skin is composed of many layers and different kinds of cells. We're only going to look at one type of cells. Cells form the many substructures of the skin, such as hair follicles, which produce each shaft or strand of hair in an animal's fur. After looking at the images above, how do you think the two different subspecies of deer mouse could have different fur colors? So here's mouse skin. Here's a close-up of the skin. You can see that each hair grows out of a shaft. At the bottom of that hair shaft, if we zoom in on that hair shaft, there are some specialized hair shaft cells that have a color to them. They have pigment in them. And we can look at those. We're going to look up at those cells up close. So now is the time to write down those ideas. They don't have to be correct. It's just your opportunity to do some thinking about what might cause those cells to be darker or lighter. What new questions do you have for the driving question board? We talked about the driving question board in lesson 1.1 and 1.2. What do you want to add to that driving question board? Here's our driving question board so far. Are mice nocturnal? What's their life expectancy? Does the fur color affect how long they're going to live? These are some of the questions we asked in the beginning. What questions do we want to ask now? Do we want to dig deeper and figure out more about what's going on in their cells? I wrote one question here, but you write your own question for question 3. Pause the video, answer the question, and then we'll then hit the next button here at the bottom of the screen. So here we have activity 4, 2.1, and we are on page 3. We're talking about melanocytes, a specialized hair cell. In the simulation below, you're going to zoom in to see the skin cells of a deer mouse that you collect from the population. We're going to explore a melanocyte, the specialized type of hair shaft cell that makes the fur different colors. By working with the simulation over the next lessons, you'll discover how the cell can produce a variety of fur colors. First, we're going to explore at the cellular level and some of the cellular structures. So here's the multi-level simulation. At the population level, we want to grab some mice and put them over here in our collection here. And then we're going to zoom in on the organism level, and we're going to see what's going on in the cell here. We can zoom even further down here on this pane. Oh, not yet, maybe on another page. We've reached the organism level. Click on your collected mouse to place it in the Explore pane. We already did that. Zoom into the mouse's skin. We did that. You just hit the plus button here. Explore the melanocyte cell. Return to the activity and answer questions. So let's just figure out what's going on here. There's some locations. That seems to be a location. Here seems to be a location. If we click on this, that seems to be a location. If we click on here, that seems to be a location. If we click on cells outside the cell we're looking at, outside the melanocyte, that seems to be a location. So there's 5 locations. Some of them look more important than others. Let's go over to the instructions here. So we've answered things. We're now down to the answer questions level here. There are 5 locations marked in and around the melanocyte cell. If you didn't find all 5, continue to explore, moving your cursor around. Choose 3 locations that you think might be important. List the 3 locations by number and suggest how each one might be important in the functioning of the cell. So let's go look at those locations for question 4. This extracellular space that's outside the cell has a bunch of different little green dots running around in there, so that might be important. The inside of the cell, the cytoplasm, always is important in the functioning of cells, so I'm going to think that's really important as well. If I click on one of these little dots, they're location 1. I can get them to show up. They keep moving. There we go. Location 1. Those melanosomes are location 1, and they're pretty important because they're what makes the cell a melanocyte, or a cell that produces melanin or pigment. Pigment is a coloring that either makes skin or fur or hair or feathers a different color based on what kind of pigment is produced. So there's some possible answers to question 4. If you come up with your own that are different, you can certainly use those. Let's move on to the next page. I'm just hitting the next button. It's out of view, but that's okay. So we've got some work to do here. We've got investigation 1. What substances are present? So we have to measure 3 locations in the melanocyte cell of the dark brown mouse to see the substances present in each area. In your field notebook, lesson 2.1, table 1, write yes is the substance present and no if it is not. So let's measure. I've done this a little bit so the graph is kind of showing here of what's where. You can click the graph here, and you can click around and use this measure tool here to go to the cytoplasm, and right now we're measuring substance D. So substance D doesn't really seem to be in the cytoplasm because that green line didn't show up here. Let's measure substance D in the extracellular space. Oh, there it goes. You can see that. Let's measure substance D in the melanosome. Not much in the melanosome. So we didn't see any green bar pop up in the melanosome here. You can kind of infer about which substances pop up in which places by these lines here. The blue line is substance B, the green line is substance D, and substance A and substance C both pop up in the melanosome. That might be a hint about what they are. This is lesson 2.1, and this is table 1. Lesson 2.1, table 1, investigation 1. This is a summary of what we found out. In the melanosome, substance A and substance C showed up, so yes and yes. I put no for substance B and substance D. In number 3, in the cytoplasm, substance A did not show up, so I put a no. Substance B showed up, so I put a yes. Substance C did not show up, and substance D did not show up either. This is basically a summary of the information on the graph we were just looking at. The extracellular space outside the cell, just outside the cell membrane, A, B, and C did not show up, but when we measured substance D, it was present, and we saw that in the graph. So the next table that you'll need is table 2 for investigation 2. This time we're going to add substances to the cell and see what happens at these various locations. Let's remember that 1 is the melanosome, where pigment is produced. The cytoplasm is number 3, and 4 is the extracellular space. That's the space outside the cell. So we add these various substances, A, B, C, and D, and we see if the cell gets lighter, which is L, D is darker, and these slashes are just for neutral. So let's go and let's try adding different things to the cell and see what happens. So I'm going to add substance D. Things change in the melanosome, but the cell doesn't get noticeably darker. So I'll add it in the extracellular space, and then I'll try adding it in the cytoplasm. Let's try the extracellular space again. Well, we did see the melanosome get the substance C really increase here. Let's try substance C. Let's add that to the cytoplasm, and let's add it to a melanosome. Ooh, that got nice and dark. Let's add it to the extracellular space and see if that does anything. Not really. Now let's try substance A. If I add it to the cytoplasm, not so much. If I add it to the extracellular space, the cell doesn't really get darker. But if I add it to a melanosome, it gets lighter. Look at that. That's pretty funky. So that was substance A made the whole cell lighter if I added it to the melanosome. Let's do substance B. So if I put substance B in the cytoplasm, ooh, that has a big effect. How about if I put substance B in the extracellular space? Not so much. How about it if I put it in the melanosome? Nope, not so much. So I collected that data in the table we were just looking at. You can try adding different substances to different parts of the cell as well and see if the cell turns darker or lighter. Remember, when we put substance C in the melanosome, we added that, the little add button, boom. So that's what darker looks like. That's what you're looking for. And when we put substance A in the melanosome, Boom, the cell got lighter. So that's what you're looking for with lighter. So that's investigation 2.